The glycoprotein Miraculin is derived from a berry, Syncypalum dulcificum, or miracle fruit, discovered in West Africa in 1725. Miraculin causes acidic foods to produce the sensation of sweetness. What we think of as the sensation of a flavor is actually a combination of two factors: true taste, which is sensations, sweet, salty, sour, and bitter, evoked by solutions in the mouth that contact receptors on the tongue and the roof of the mouth, and then connect to axons in cranial nerves 7, 9, and 10, and retronasal olfactory sensation, which is the sensation of an odor that is perceived when chewing and swallowing force an odorant in the mouth up behind the palate into the rose. Such odor sensations are perceived as originating from the mouth, even though the actual contact of odorant and receptor occurs at the olfactory mucosa. The act of tasting is broken down into several steps. Chewing breaks down food substances into molecules, which are dissolved in saliva. The saliva-borne food molecules flow into a taste pore that leads to the taste buds housed in structures called papillae that are located mostly on the edges of the tongue in a rough oval. Taste buds contain multiple taste receptor cells, each of which responds to a limited number of molecule types. When it contacts with one of the preferred molecules, the taste receptor cell produces action potentials that send information along one of the cranial nerves to the brain. Humans are capable of perceiving four basic tastes: salty, produced by the cations of salts, e.g. the sodium and sodium chloride; sour, produced by the hydrogen ions in acidic foods; bitter, produced by substances like quinine or caffeine; and sweet, produced by some sugars such as glucose, fructose, and sucrose. In the process of sensing a sour flavor, hydrogen ions enter the receptor cells through ion channels. The undissociated acidic molecules dissociate inside the cell. Ultimately, the stimulus that triggers sour taste is the proton concentration inside the receptor cell. It is evolutionarily advantageous to be able to sense sourness, as it helps animals detect highly acidic compounds. Mildly acidic chemicals producing sour flavors, however, can be enjoyable. The taste receptor for sweet compounds is a heterodimeric G-protein coupled receptor consisting of two subunits, T1R2 and T1R3. Both subunits consist of 7 transmembrane domains. Sweet receptors can bind a variety of ligands, including sucrose, fructose, aspartame, and saccharin. Sweet receptors are localized on the extracellular side of the plasma membrane of gustatory or taste cells. Upon binding a ligand, sweet receptors activate their associated G protein, gusducin. Gusducin, in turn, activates adenylate cyclase, which results in upregulation of cyclic adenosine monophosphate. The increased concentration of CAMP activates potassium channels in the taste cell membrane. The resulting efflux of potassium causes the cell to depolarize, triggering the activation of calcium channels in the ER. Finally, this spike in calcium causes the taste cells to secrete vesicles containing neurotransmitters targeted at an associated neuron. This signaling process produces the sensation of sweetness. This is a schematic for a proposed model of Miraculin's mechanism of action. First, Miraculin binds to the HT1R2, HT1R3 receptor when it makes contact with the tongue. It stays bound for up to an hour. When a sour solution is introduced to the bound Miraculin, Miraculin acts as an agonist. Miraculin then reverts to an antagonist when the local pH returns to neutral, preventing other sweeteners from activating the receptor. Protonation of the histidine residues in Miraculin, histidine 30 and histidine 60, are key steps in making Miraculin an agonist. Since Miraculin can fluctuate between agonist and antagonist, the protonations most likely change the conformation of Miraculin, not its affinity for the receptor, thereby activating the receptor.